So I think we'll get started at the official 10 after, or 8 after. Welcome everybody. Big day for Daniel. And as the tradition goes, I'm Sergio Introduction, I'm his advisor, and I'm going to introduce Daniel. Daniel was my first graduate student, and I can confidently say that he was the best. Working on them all. Seriously, I think he really set up a high bar. I mean, I don't know if all of you guys in the other labs are like him, but he set up, and this is for the ones that come after him. It will be tough to fill his shoes. But I would like to make a few remarks about his personality, his style. Daniel is certainly open-minded. He's open source. He's open everything. He set up a new standard in the lab, shared everything. There are a few things that I wish I didn't know about him, but very openly, very so much so that, and he had some particularities. He's one of the few people that I know, actually the only one I know, that wasn't comfortable with publishing in Nature. He had material for Nature, but he said, "Nah, I don't think because they're not open source." And okay, so he got his way. He published in PLOS Computational Biology. But the funny thing is that actually Nature published him. And I don't know if you're going to tell the story, but okay.
You can tell part, yeah,
I'll say no more. I'll say no more. You break it. But so yeah, he's particular in many ways. UCSF is a tough place. Admittedly, it's a cruel place. This is what comes in as a very young fellow, just out of college, so full of energy and young looking. And I think this is what comes out. Yeah. So actually he was smart. About a year and a half before he graduated, he started growing his mustache. And he said, "I'm going to shave it when I graduate." Gee, I was looking forward for him to graduate. So, but anyway, for the ones who come after him, that trick won't work anymore. So try another one, particularly girls. So, this is Daniel's habitat. When he started in the lab, we have a hybrid lab, so half wet, half dry, and I had a spot in both the cubicles and the lab. And I offered him, "What do you want? Where are you going to go? You want to go to the cubicle or the lab?" "Oh no, can I go to the lab?" "Ooh, let's go to the lab." So he went through all the training, unnecessary, by the way, but he wanted to be there, and he felt at home. As you can see, he decorated it to his liking. He even had a few things, I don't even know what they are, in there. So, what's this little thing?
Oh, it's my weather station.
So radio don't work?
Yeah.
Okay, so and this is his plant. Look at his plant. He even had a plant there. And then I saw a little number down here. I said, "Oh wow, this is a serial number for the plant?" And then I looked closer. What kind of plant is this? Daniel. Not only that, then he's also using in the lab. This is a lab, Daniel. Alright. So, okay, fashion is not his thing. So he has many talents. Fashion is not one of them. But apparently this runs in the lab. Look at this. Funny combination. I guess I didn't set up a good example here. So, he has other talents. I don't know exactly how the story goes, but you have to see this. Unbelievable. Really good at it. And look what he did next. There's a lot of stories to tell. So when Daniel had his first committee meeting, he laid out his brilliant plan, and this is what I want to do, and the possibilities when I graduate. You can see down there. It could be a postdoc, it could be the industry, it could be an independent scientist, it can start up a company. That was his first meeting. And then he said, well, yeah, let's go down to earth. I really like machine learning, and I want to do something, and I like networks, and I want to do something with networks. And I say, okay, that sounds great. And I say, yeah, I really like machine learning. But then, seems like the machine will learn. I don't know what you were doing out there, but that was interesting. There's a couple of explanations that deserve, I think. Like for this one. I know the story, you need to tell people what this means. And this. So I think that there's a lot of stories to be told. I'd rather mention one very interesting. He is so open science, open everything, that he even announced his decision to graduate on the internet. Decided the future of Daniel. No. You have to listen because something happens in the first 2 seconds. Let's see who picks it up.
Welcome to St. Malo, France, on the Pacific Ocean.
He sent me this and I was traveling and say, oh no, I'm jet lagging. You were jet lagging. Oh my god. Anyway, I think stories need to be told. I thought we should end it on a high note. I think you really did a great job. You were in the, you got your summit beautifully, and we're all looking forward to your exit talk, and best of luck. With this, I would like to give you the inaugural Baranzini Lab graduation kit.
Wow.
And a complete mate set.
Ah. Fantastic. That was a mate gourd, not a piece we were drinking out of. For those who are curious. Okay, first, thank you, Sergio, for those kind words. It's really been a pleasure working together, and I see a lot of familiar faces here. It just makes me so happy everyone who came. So I will be talking to you today about what I've done over my thesis. I've titled the talk, The Hetnet Awakens, Understanding Disease Through Data Integration and Open Science. And for those that are curious about what the background image is here, this is what happens when you use deep neural networks to extract the style of Dipper, which was my PhD workstation, and you combine it with my avatar, and then you get that amalgamation. So this is the cutting edge of machine learning. I thought I'd make a cool picture. And I guess Sergio needs no introduction, but it's been fantastic working in the Sandler Neurosciences Center in the Berenzini lab. And my PhD is in biological and medical informatics. So let's begin. Back when I was starting graduate school, I had heard a little bit about networks. Networks are just entities connected by relationships. And one question I asked, or just one simple exercise, was what can I tell about my Facebook friends knowing just their mutual friendships? So I constructed a network built from my Facebook. This was back in 2012. And each dot here is a friend that I have on Facebook. So if you have more mutual friends with me, you're a bigger dot. So Willie, for example, who went to high school with me, who's sitting in the back there, is probably one of the bigger dots, like this one here. And all the edges here, or what we call relationships, are when two people have a friendship. And so just knowing who my friends were and which friendships they had between themselves, you can get an incredibly reliable picture of the different communities I've been a part of. So just using a layout algorithm that uses the information on friendships, you can split the individuals on whether they're from my time at Cornell, whether they're from my time at Cornell but from the debate team, because it was a very separate community I was in, or whether up here they're in this extreme that's my kin, my mother's family. She has a really big family, so that's why there's so many, or whether they went to summer camp with me. And so I got interested, could we apply some of these same principles to biology? So with network biology, rather than friends, we're looking at potentially diseases or genes and trying to understand similarities between them. And one of the first projects I did was looking at the genes associated with disease. So here, the bigger circles are diseases. So for example, ulcerative colitis, multiple sclerosis, breast cancer. And then these smaller circles are genes. And the lines represent the relationship when a disease is found to be affected by mutations in that gene. So whether disease susceptibility depends on that gene. And what you see is many genes, are like this one, are only associated with a single disease, but some are associated with multiple diseases. And so the question became, can we create a network which shows which diseases are similar to which diseases based on, I guess, the shared variants in the genome that cause susceptibility. And so when we did that, we found something interesting. In general, diseases would cluster by their type. So purple here represents autoimmune diseases, which are generally pretty close to each other. The solid cancers as well also cluster together, a little bit less so. And then we have 3 blood cancers. One of the blood cancers, multiple myeloma, looks a lot like a solid cancer. Another one, chronic lymphocytic leukemia, looks like it has a genetic architecture that's in between solid cancer and autoimmune disease. And then Hodgkin's lymphoma, which the study was about, looks a lot like an autoimmune disease from its genetics. So this was interesting to us because that potentially could inform different methods going forward for how to treat and study Hodgkin's lymphoma. However, we felt we were making some oversimplifications. We were unsatisfied that we had to condense everything onto a network of a single type of entity and a single type of relationship. So here everything is a disease, and we took information about genetic variants and we collapsed it and projected it onto a single type of relationship. And we thought reality is much more diverse, that there are many different types of entities at play in a biological system. Can we capture that? So, we began working on Hetnets. Hetnet's a term that may have existed before us, but we are definitely trying to popularize it, and it is short for heterogeneous network, which is a network with different types of nodes or relationships. The first project I worked on, which we can call hetio disease genes, was to predict when a disease was associated with a gene. So that's the background of this last slide. Can we predict that using the other parts of the network? So here we have our diseases, about 100 complex diseases, and all 20,000 protein-coding genes. And these edges between them are the associations that have been collected from human genetic studies over the last decade. So what we did is we built this network to integrate diverse data to potentially give us a better idea of why these relationships exist and why other ones do not exist based on the context of the rest of the network. So the network in total has 40,000 nodes of 18 types and 1.6 million edges of 19 types. Now, the way we approach, theoretically, this problem is by looking at the type of the graph. So here we have a bird's eye view or schema of the network, and we have our genes, which are associated to disease, and we're interested in different ways that these two entities can be connected. So we define something called a metapath, which is types of ways on this network to get from a gene to disease. So one example would be that a gene is expressed in a tissue that is affected by the disease. And for this project, we went up to length 3. So for example, this shows when a gene has a protein interaction with another gene, there's a protein interaction with another gene that's associated with the disease in question. And we created a framework called the degree-weighted path count for any gene-disease pair to quantify its connectivity in the network. So if you gave me a gene and you gave me a disease, I could give you a bunch of what we call features or numbers back that describe the relationship. And our goal was to learn which of those numbers actually mean anything and predict whether a gene is associated with the disease. So the way we do it is for a certain type of path, for example here, whether a gene is expressed in a tissue that is affected by a disease, we find all the paths. Here there's one. And in some cases we have multiple paths, like here for gene interacts with gene which is associated with the disease, we have 3. So ERF1 interacts with IL-2RA, which is associated with MS, and we have 3 of those connections. And then we perform a weighting to downweight paths that go through really connected nodes, which tend to be less informative. But in the end, here's what we found. We were able to look at paths that go through different types of gene sets, so different types of ways of understanding what a gene in a human does. So this is for a path where a gene is part of a gene set that another gene is part of that is associated with the disease. And we found Reactome here, which is trying to encode human pathway knowledge, which is very commonly used for similar approaches to prioritize genes for association study, performed okay. The higher you go on this axis here, the better performance you have. However, perturbation gene sets, which were a high throughput measurement from differential expression experiments, that sounds complicated, but it's just a really high throughput way to look at what happens to cells when you perturb them, say by giving them a chemical or altering them genetically. This didn't require any expensive curation like a lot of the pathway databases. It was sort of all automatic in a way, and it produced very high predictiveness. But in the end, our model, which combines all the information, outperformed any individual type of information, and that's our motivation for the project. So what we do is we fit a model which looks at the different types of paths and combines them into a prediction or a probability that a gene-disease pair is an association. And we showed that we were able to do many cool things, one of them being if we hide a certain study, say this one on multiple sclerosis, we can predict what the genes will show up. And we use this ability to identify 4 new genes, which then also were significant in a validating gene loss. And we put the predictions online, although they don't seem to be loading, so we'll skip this. So the next project I did, which is sort of the final act of my dissertation, is called Project Repedio. And whereas my last project, it was just really Sergio and I doing it, this has been a massive collaboration. I'll get to that more later, but I just want to thank everyone on our team. And the goal here is to use the same technique as before, but now predict which drugs treat which diseases. So the goal essentially is to understand why drugs work on a large scale. So systematically across all small molecules and all complex diseases, why do the drugs that work treat the diseases that they treat? So this was actually my first project in Sergio's lab. And after my qualifying exam, which I did this on, I did the disease gene project, and I got back to it. So in 2013, this was the schema for the network that I planned to create. And now here in 2016, it looks like this. So we've had a lot of growth in the network, which I'm super excited about. We call the network Hetnet. And some exciting types of data which have just come out are whether a disease changes the expression of a gene, and say whether a compound changes the expression of a gene, so whether it makes more or less of it. These are really high throughput measures. We also have whether compounds treat disease or whether they palliate it, which means treat a symptom. For this, we had to get 3 physicians to go through 1,300 automatically extracted treatments because a lot of them actually were symptomatic. So I'm really thankful for these physicians who spent their time to tell us what treatment was symptomatic. So for example, we had cocaine treating cavities, which is most likely a symptomatic treatment because it's used as an anesthetic. So that's not something that we would want a teacher approach to predict. So that's why we have the doctors. So this is what it looks like. Hetnet version 1.0, just released about a month ago. And it's super exciting. It has 1,500 small molecule compounds and 137 diseases. Between them, they have 755 treatments. So this is what we want to understand, why those treatments exist. And so we built the rest of the network. We have entities such as side effects, which could be interesting because what's a side effect in one application could actually be useful for treating another disease. And we have pharmacologic classes of those compounds. We have the symptoms that diseases are part of. We have anatomies, or the tissues, that a disease affects. We have the protein coding genes laid out here, all 20,000, and then different gene sets. We don't have as many gene sets as before because we found they were redundant in the information they gave. And so overall the network has almost 50,000 nodes of 11 types and over 2 million edges of 24 types. And together this was from 29 different resources that combine millions of studies from the last half century. So what's exciting to me is we've taken all this knowledge, and sure, we've made some simplifications, but we've been able to put essentially a large part of the last half century of biomedicine into a single Hetnet. And it's super connected. At the beginning, so if you're only looking at single edges, a lot of the different types of nodes you can't get to from each other. There's no edge between them. So it's colored in here if there's an edge between them. But as you increase your path length, you start getting amazing connectivity. So if you're looking at paths up to length 4, there are about 3,000 ways to get from, say, a gene to another gene. 3,000 different types of connections. Each type of connection, or each type of path, encodes a different aspect of information. And so that's exciting that we are getting incredibly diverse, potentially, and detailed descriptions of how 2 entities are related. And so we have a machine learning paradigm where we have 1,206 types of paths of length 4, and we have about 200,000 potential treatments, so potential pairs between compounds and diseases, things that we want to rank and say, is this a good pharmacological opportunity? Should potentially pharmaceutical companies look at this for repurposing? One aspect of our approach is that it now takes drug companies over $1 billion in research and development to discover a new therapeutic. So can we make this a lot cheaper by taking existing medications which have a known safety record and are already approved or already used, and then use them for new applications, because oftentimes they have multiple effects. And so we have 6 different networks. We have to make 5 permuted networks to sort of control our experiment. So this means we have to do about 60 million database queries. So it can take a lot of time. We have to do it in parallel, and it's been exciting learning the new technologies to do that. But in the end, we get a model which predicts whether a compound will treat a disease, so it gives you a probability. And our predictions are looking good, but they should become really finalized in the next few weeks. So now, the next project is a little project I did, which was super easy once we had constructed the Hetnet. And ever since working at Princeton in the summer of 2012, I had this idea about something called the SNP plentiful effect, which was the idea that when genes have more genetic variants, more things that vary between humans, that they potentially have more connections in the network. And this can cause biases in a lot of scientific approaches, or a lot of network biology approaches. So we think that potentially a lot of network approaches are seeing results which just come from this sort of non-random distribution or non-random association here. And what we saw as the number of SNPs in a gene increases, in general for these important types of network edges, the number of connections in the network increases. And this was true on genotyping platforms and sequencing platforms as well. So now I'll get on to another study, which is a little bit separate from the rest of my work, but I would say one commonality is the statistics and the fact that it relied all on public data. Actually, my entire dissertation relies only on public data, and that's even now an explicit choice with how I want to proceed. So my roommate at the time, Kamen Semyonov, who many people in this room will know, had this idea from looking at cancer maps that there was less lung cancer at higher elevations. And his hypothesis was that at higher elevations there's thinner air, there's less oxygen, and potentially breathing the oxygen is actually carcinogenic. And this does have biological support in that oxygen is known to create free radicals. It's highly reactive, which is one of the reasons why life has been able to harness it so effectively. And so we decided to limit our analysis to the western United States, where there's a lot of elevation variation. So just this part right here. And what we found was this negative correlation between the elevation of a U.S. county in the west and its lung cancer incidence, which is here on its y-axis. So just to show you, San Francisco is this dot right here. We are at near sea level, and we have, for that elevation, slightly lower lung cancer incidence. But one thing here is that in this plot we haven't adjusted for all the other confounding factors and potential other reasons why people get lung cancer, foremost amongst them smoking, and other demographic variables that can be proxies for a lot of different health behaviors. So what we did is we looked at, I showed you before the correlation between elevation and lung cancer. But now we collected many other variables. So for example, here we see the correlation between smoking and lung cancer, and as you expect, it has a red correlation or positive correlation, whereas elevation has a blue or negative. So we also had to look at how all the different predictor variables, or all the different variables we assembled were correlated to each other, and you see a lot of these variables are correlated, and we have to take that into account. Luckily, the modern regression models are able to do this. And to control the analysis, we looked at 3 cancers, colorectal, breast, and prostate, that don't have direct atmospheric exposure. So our hypothesis there being that we will see less of an association with elevation because they don't have direct exposures. And what we found after accounting for all these different variables that we included was that for lung cancer, we have these highly negative associations between elevation and lung cancer. But for the other cancers, we don't have those associations. So, to sort of summarize the project, this is San Juan County, Colorado. This is a picture from 1908 when the bell tower collapsed while it was under construction. But this is the highest county in the United States with an average elevation of 3,400 meters. And it has, compared to where we are now, 35% thinner air and therefore 35% less oxygen. And we estimated that if all of the United States were to elevate to that level, there'd be 65,000 fewer new cases of lung cancer per year. Obviously this...
Let's do it.
Yeah, let's do it. So, one thing I would be frustrated, as my time during a scientist, is it seemed like science could be done way more efficiently. And here we have these scientists dragging a cart with square wheels. And someone in the back has these wheels and they say, "No thanks, we're too busy." And I see that a lot in science, that we're doing such incredible work, we're so focused on our work at hand that sometimes we may be missing massive ways to improve our efficiency as a whole. And This didn't show up as I expected. I guess Unicode is still not fully standardized, but there should be 68 of these hourglasses. And what they represent is when Sergio and I had our first paper accepted to PLOS Computational Biology, we had waited 68 days and it still wasn't published. And after acceptance, the paper's not changing at all. It's not like there's the peer review has already been done. So it's really just time where your work is not getting out there, not getting the attention that you want it to have. And so I got really frustrated, why is it taking so long? What should I expect? So I ended up looking at a bunch of journals in my field and finding the distributions of their delay times, because the journals actually report the timestamps in the articles once they're published. So they report the date that they were submitted, the date they were accepted, and the date that they were published. So date published minus date accepted gives you the distribution for each journal of the delays. And we saw there was a wide variation. Some journals published really quickly after acceptance. But the two that Sergio and I had submitted to were the slowest of all, so I guess that's our luck. But I guess this was the story Sergio told at the beginning. I said I didn't want to submit this study to Nature Genetics because I didn't have a CC by licensing option, which is open access so anyone can read it. But this caused a big stir on Twitter and everything, and they promptly wrote an article covering these findings, mentioning the article. So Sergio finds that funny. Oh, paradoxical. So then I did another project about 6 months down the road where I expanded to looking at the time from submission to acceptance. So this we can call like the review time or acceptance delay for all articles in the PubMed database. So before we were looking at just from 2014 on, but now I switched to 3 million articles since 1965. And what we're going to see is whether acceptance delays have gotten worse over time, which a lot of people think they have because they're so frustrated with the process. Uh-oh, we've hit a paywall. So for those who are not familiar with paywalls, a lot of scientific literature, even though it's publicly funded, you get to it and it wants you to pay. And this is actually the first recorded or documented paywall from Science magazine in 1997 from the Internet Archive. But as Mike Eisen points out, it was a lot cheaper back then, only $10. You're looking at a heftier price tag. But I can remove the paywall. So what we see is about a median time from acceptance to publication of 100 days. And over time, we found that that didn't really change. So maybe journals aren't actually getting that much slower, but there's still a lot of room for improvement. And one interesting outcome here is that this is a journal PLOS Computational Biology, and what's blacked out is the time after I made my initial blog post, in between this next blog post. So we're going to get to see whether my blog post potentially had an impact on their times. And we see a drop-off, I can't promise that I was the cause of this. I guess, to me, I think transparency in the publishing arena can help with a lot of these problems, because right now it's unacceptable. That brings me to the grand experiment, I would say, of my PhD, which was, for the last project, we've been posting our results and our discussions on it in real time. So as soon as we have a thought or get around to writing it, we post it online on a site called ThinkLab. So this is our project. We started from when it was a proposal, and we now have 40 different contributors who have come from all different places. Oftentimes they were the leading experts on a certain database that they had created. Over 71 discussions of content, and 424 comments. What's cool here is that all these discussions have about 83,000 words. We're approaching 1 million characters in all these discussions. And if you look at the average length of a publication in the literature, we've made about 14 publications before even finishing our project in worth of words. So I actually think it's valuable content, but I just want to show that a lot more scientists have a lot more that they're not putting out there that they could. And since starting this in about January of 2015, my GitHub public contributions have really taken off. So it's also spurring me to make a lot of code because I have to reference whatever I'm saying and support it. And here's a beautiful graphic made by Antoine Lizet, who's in the audience, which shows the growth of different users over time, weighted towards new users, but it shows that we've had a lot of different contributions. And here I'd like to highlight a few all-stars who really, I think, are awesome examples. So the first, the format I'm going to do here is show the person and then their comment, and then I will reveal the result after their comment. So Lars Jensen from Europe says, others like myself try to remember to rate everything that I've read. So actually I tweeted him and I told him about our project, and then he went through and he read every single comment, even ones that were months old, and rated every single one of them. So that was really amazing to have just someone totally uninvolved with the project come and show so much interest in it. Next, we had some legal issues, which I'll get to, and Katie Fortney starts off by saying, I am a lawyer, but not your lawyer, or UC's lawyer, and this isn't legal advice. And then proceeded to give a 1,500-word post of some of the best advice. Cocaine was not disease-modifying for cavities, and he created almost 3,000-word posts on his exact methodology. This is really valuable because there wasn't a public resource that had this information that was physician-curated and openly licensed before this. And then we have Antoine commenting on the math feature, where he said, it took me a while to figure out. And then after that, he started making formula after formula after formula after formula. And I hope he has many formulas left to come. So here I'll talk a little bit about the legal issue that I was very excited when we released a preliminary version of our network. I called it One Network to Rule Them All. And I said we've completed an initial version of our network. Network existence is proven in Bitcoin block 369,898. So I actually took a fingerprint and put it in the blockchain because that's the most immutable way to store something that people know about. I was excited about that. And then Lars said, nice of you to share this big network with everyone. However, I think you need to take care not to get yourself into legal trouble here. I'm not trying to cause trouble, just the contrary. When making a meta-resource, licenses and copyright law are not something you can afford to ignore. I regularly leave out certain data sources from my resources for legal reasons. And we ended up having a lot of discussions on this topic, but the background of this slide, I've wrapped a single discussion, so it just shows you how much effort we had to put into sort of understanding all of this, because these topics are complex, and it's an area that the community hasn't really addressed properly until now. So there are legal barriers to data reuse, and they're arising a lot more as people start using more data that they didn't create, which is a more recent practice. Our network, Hetnet, integrates data from 29 resources. Only 12 of those had a license that met the open definition. So a license is something that's put on a resource and tells you how you can use it, what you can do with it, and gives you certain permissions that otherwise the default copyright protection prevent. 9 had no license. If a resource doesn't have a license, that equates legally to all rights reserved. Now, a lot of scientists put their data on the internet assuming that once it's available, it becomes reusable. And in their mind, it is, but legally, that's not the case. And there are also incompatibilities between licenses. So for example, if one license says the data can only be used non-commercially, and then another license says you need to share it alike, but doesn't have that non-commercial stipulation, those two potentially can't be combined. So a lot of our work had to deal with how can we deal with this. And for 11 resources, we ended up requesting permission from the creators, or who we thought were the copyright owners. And this is what you have to do if you want to use a resource that does not have a license. The median time to first response was 16 days. And so when you're integrating a lot of resources, this creates huge delays. And only 2 of the people affirmatively responded. No one said we had to take their resource out, but the main issue is they themselves were confused. They didn't know whether they owned the resource, whether the university owned the resource. They had often integrated other resources that had unclear licenses. So it's a big problem. And we ended up removing MSigDB, a project from the Broad Institute with lots of public funding. I'll just mention here that all of the projects that we used were publicly funded, and essentially by organizations with the purpose of advancing science. So it's not like we're taking data that wasn't meant to contribute to science. And their license, which you have to agree to make an account and download the file, says, licensees agrees not to put the database on a server that may be accessed by any individual other than the licensee, which I think would have, almost all of our servers, would violate that policy. And I think this is an example of when a legal department at the school sort of runs amok of the scientific intent of the funding. The license goes on to say, licensee agrees to provide a written evaluation of the program and the database, including a description of its functionality or problems and areas for further improvement. So when you're integrating 29 different resources, you just can't have each one of these having these esoteric restrictions like this. So we ended up removing MSigDB, which also says you can't redistribute it, so it wasn't a question of whether we could keep it in. So my advice in general, and what the practice we're trying to abide by, is to release data under an open license. You can go to this link, and there are about 5 conformant licenses with open data currently, but the one I use, which is the public domain dedication, is called CC0. And finally here, we have a very special component of the presentation. Let's see if this works. This is Latin for acknowledgements. I wouldn't be a PhD without any Latin. So first I'd like to thank the NSF, who gave me a fellowship, which supported a lot of my graduate study. They have a really specific way that they'd like to be thanked, which I put up here. Specifically, they want me to say, any opinions, findings, and conclusions or recommendations expressed in this material are those of the author and do not necessarily reflect the views of the National Science Foundation. Next, I'd like to thank the open source scientific software stack that made my whole PhD possible, and probably most of the other PhDs in this room. It's really an amazing thing what open source software has done, and probably most of you use it without even knowing it. And also the free scientific tools that I have used and have been extremely helpful. And foremost among them, obviously, ThinkLab and the founder, Jesse, who really tailored the service for our needs and was responsive to helping us do a real-time science. The next acknowledgement here, I would like to give to all the journalists who've taken the time to understand the work. And I find they actually, sometimes the journalists spend more time than scientists digging into the work and trying to understand every part. And there's 4 that come to mind here. Kristen Sanini, writing for the Biomedical Computation Review. George Johnson, writing for the New York Times. Kendall Powell, writing for Nature News. And Simon Oxenheim, who should be writing for someone, but still has to publish an article. But he spent a lot of time understanding the legal issues surrounding our data integration, so I hope that goes somewhere. And then, of course, Cayman and Kattini. Cayman's cat I lived with for several years, and I will rejoin in the next step. And this is for the Pure J Holiday Video, which is a journal. I'd like to thank them too, because they're really fantastic with their lung cancer study and helping us out and helping with the press release and stuff. And then finally here, this is my brain, but it descended from my father, Marty, who's in attendance, and my mother, who's not in attendance. But I want to thank them both incredibly. One thing is that the summer before coming to graduate school, my mom was diagnosed with multiple sclerosis, and so it's been incredibly special for me to be able to work with Sergio and the rest of the MS team here at UCSF, one of the leading research MS teams in the world, and understand it all. Luckily, she's doing well, and my brother as well, who provided the important environmental influences at the top of the genetics. The brother's the environment. And I'm playing around with a new way of acknowledging family members. So here, this is my 23andMe results. And we have the blue sections are the ones that my maternal grandmother passed down to me. And here are the genes, or the locations or variants that are associated with educational attainment. So my PhD ranks these genes. And then my great student mentors or colleagues I've had here, Cyrus Maher, Nina Gonzaluto, and Mason Louis, who've taught me so much, and then also the people I had the pleasure of mentoring here. One of them, and the first, being Maria, who found Sergio, and actually came from Guatemala. She had a goal of curing ALS, and Sergio said, come on in. She actually was only 15 when she came, so a really remarkable story. She was so dedicated. Leo Brugman and Sabrina Chen, all fantastic, and it was great to be taught and to also teach. And then here, there were a lot of friends, so I had to pick 2 highlight photos. This is Graham at Billy Goat Hill overlooking the city. So 2 birds with 1 stone, Graham and the skyline. And then, we had a lot of fun in the rear view. It was a great time. Here, you can see us out. And then, of course, the lab. Observe everyone in this photo. Here we have Sergio, Antoine, Egle, Sherman, Sneha, and Kitchell. It's been great. The current lab, this is at our ski retreat. And then there's been so many other people who have been in the lab over the time, so I've put them all here. There's a lot of them. And even more, it didn't fit in my diagram. The different faculty that we have lab meetings with, and everyone who's participated in the group, which has been fantastic. And finally, Sergio, what a great experience we've had, what a fun time. I'm so happy with how everything worked out, and thanks so much for it all. Now, this is Sergio in the lab. He's mostly computational these days, but he will come in and do some wet lab from time to time. So this is him with Egle, but actually I wasn't a big fan of when he came in to do wet lab, because one of the main reasons was he needed to take my blood for the experiments. So that's him there, extracting my blood for Egle's microbiome study. And finally, I will acknowledge all the academic mentors I've had from this point. So it was the summer of 2007. I had just graduated high school and I was unemployed. So I emailed Jason Moore, a professor at Dartmouth, and he said, come on in. And I was like, oh, that was easy to get a job. But a few years later, I said, Jason, what's your key to success? And he said, well, I say yes to everything. I now understand. And Jason had a graduate student, Casey. Casey ended up doing his postdoc in Princeton with Olga Troyanskaya, and when he moved to Princeton, he got me the summer job there with Olga, which was fantastic, and I learned a lot. And then after that, I came to UCSF, and around this time, I had my rotations. So Sergio, John Whitty, Ryan Hernandez, and my PhD here from 2011 to 2006. No, my Quals committee in 2013, Andre Shawley, Katie Pollard, Patsy Babbitt, and Jun Sung. And then my thesis committee, Andre Shawley, John Whitty, and of course Sergio as the chair. And at some point during my dissertation, I became D. Himmel and got a new avatar. And during the time I've been here, Jason and Casey actually both moved to Penn. And I'm excited to announce my next step. We'll be doing a postdoc in the laboratory of Casey Green at Penn. So I'm excited for that move. And here's the grand ensemble of everyone who's mentored me. It's been so fantastic. Obviously, I couldn't have done it without these people. So, that's it. So, you can find the presentation online. If you're interested in parts, you can click on it, and I'll bring you to the link. We have a reception and exhibit to follow in Sandler Neurosciences Center. I have arranged an exhibit and I'll also present Sergio with 2 gifts. I know at least one of them he'll really like. And if there are any questions, I can take them now. But if there are no questions, we can all go celebrate. So, thank you, everyone.